Is it still not visible?
It's visible, sir. It's visible. You can start.
Yes, yes. Good morning, everyone. I'm so sorry for the technical issues there. Uh, and at the outset, I would like to thank Dr. Shinoy, Dr. Uma Maheshwari, the Association of Fungal Biologists of India, the Association of Microbiologists of India, Mysore chapter, and the JSS Academy of Higher Education and Research. So I have a disclaimer before I start. I am just the new kid on the block. And my teachers, Professor Arunalokei Chakravarti and Professor Shiv Prakash Rudramurthy, are the stalwarts in medical mycology in India. And I can proudly say that I am a satellite colony of PGI Chandigarh, and we are trying to help people with fungal infections here at AIIMS Bhubaneswar. So, proceeding further, in 2019, there was an article in the European Scientist which said that only one fungus was responsible for 90 species extinctions. Imagine the impact of fungi on ecology, right? So, based on this and inspired by cordyceps, there was an episode in the series The Last of Us which said that we are nearing a fungal zombie apocalypse. But then on the same year, in the Mirror, there was an editorial that the the fungi converting animals into zombie, though it is true, but humans have a far bigger problem to deal with it. So what is the problem? The problem was that in 2023 alone, there were around 6.5 million serious fungal infections, with more than half of them leading to the death. If you looked at the number of deaths due to fungal infections, what was the number? The number was around 6.7% of all global deaths. This surpassed the deaths due to tuberculosis, malaria, hepatitis, or pneumonia, which receive massive funding from the funding agencies, right? So back in 2017 itself, there was an editorial in Nature which said, "Stop neglecting fungi." Now, why this was said was because the editors then themselves realized that fungi are virtually ignored by press, the public, and the funding bodies, despite significantly threatening the public health, food biosecurity, and biodiversity. This was long before the concept of One Health, which was introduced by WHO in 2023. So, while this came back in 2017, it took a while for the funding agencies, the major funding agencies, to realize this. So, what made them wake up? It was this fungus, a fungus that quietly swept the globe. And this was Candida auris, a fungus that was isolated from the ear of a patient with otitis media in 2009, and in a decade it spread to all the continents of the world except Antarctica. Now, why the healthcare community is scared of Candida auris? The healthcare community is scared because of Candida auris, because patients in healthcare establishments can acquire this infection and can transmit this infection or can transmit this bug very fast. It is said that it takes 4 hours for the fungus to get colonized in the patient's body. And this character allows it to spread rapidly within the healthcare environment. And what's more worrying is that Candida auris is highly drug resistant. Now, this was a paradigm shift in Candida infections, because here we had a yeast that was acting like a bacteria. So 2019, CDC woke up and said that drug-resistant Candida are a big, serious threat to human health. But then the sibling of the yeast, the fungus, the mold, it also made its debut. And there were publications, a lot of publications, which said that agricultural use of fungicides was resulting in azole-resistant Aspergillus fumigatus infections, which are a major cause of human infections, right? And this actually woke up WHO, and WHO released its first ever list of health-threatening fungi in 2023. So by now, we know that fungi can cause deadly disease and can develop resistance to antifungals. So what are antifungals? Now, antifungals are drugs that kill the fungi or inhibit their growth when given to a person with fungal infection. So mainly the four major classes of antifungals include the azole group of drugs, the echinocandin group of drugs, the polyene drugs, and the nucleoside analogs. These are the four major groups of the antifungals that act against the fungi. Of these, the azole group and the polyene group, they damage the cell membrane of the fungus, leading to its death. Again, echinocandins also inhibit an enzyme called as beta-glucan synthase and result in death. While nucleoside analog 5-flucytosine damages the nucleic acid and causes fungal death. So why should we worry about antifungal resistance? Fungi cause death. But why should we be worried about antifungal resistance? Is because if you look at this is a table listing the classes of antibacterials and the number of antibacterials that are available. And this is the list of the antifungals that are currently available for treatment of fungi. If you can see, the total number of antibacterials in one group, that is a cephalosporin groups itself, exceeds the total number of antifungals available for treating serious fungal infections, which means that we have very limited number of antifungal drugs, which translates that it is easier for the yeast or the fungus to develop, to become pan-drug resistant, that is become resistant to all, because it has to jump a fewer classes than compared to bacteria. Also, the available drugs are highly toxic. Why they are highly toxic is because both fungi and humans, they are eukaryotes. So the targets that are there in the fungus, their orthologs can also be present in humans, and that can cause extremely bad adverse reactions in humans. Moreover, the majority of the research that is available focuses on Candida, Aspergillus, and Cryptococcus, the three main fungi that cause human infections, and other pathogens like mucorales that cause mucormycosis and the neglected fungi like Madurella fonseca, which cause maduromycosis and chromoblastomycosis, are very, very neglected when it comes to research on their treatment. So let's look into what is antifungal resistance. So before looking into antifungal resistance, we should know how antifungal resistance is measured. So how antifungal resistance is measured and what are the associated terminologies? So antifungal resistance is measured by using specific tests given by two authorities, that is Clinical and Laboratory Standard Institute of the United States of America and the European Committee on Antimicrobial Susceptibility Testing. So there are two types of testing methods. One is the diffusion methods and the other ones are the dilution methods. So the ones that the value that is obtained after testing a fungus against an antifungal is called as a minimum inhibitory concentration. Now, what is minimum inhibitory concentration? Minimum inhibitory concentration is the minimum amount of drug that can inhibit the growth of the fungus. Now, in clinical microbiology, minimum inhibitory concentration is used in conjunction with breakpoints. What are breakpoints? Now, breakpoints are values which are again given by these two organizations. And if a fungus has an MIC below the breakpoint, it means that it is called as a sensitive isolate. And it means that if we give this drug to this patient, the patient will respond to the antifungal drug. And if the MIC is more than the breakpoint, then the isolate is considered to be resistant and there would not be a favorable response if this drug is given to the patient with the fungal infection. Now, in research, the definition of resistance is straightforward. It is an isolate is said to be resistant if its MIC is greater than the MIC of a reference isolate. However, it is not as easy as has been showed there. That is, if the bug is sensitive to the drug, it will respond. If it is resistant to the drug, it will not respond. Antifungal failure in patient can be a result of clinical resistance, which again is dependent on the patient factors, because it's not only the drug that helps in taking away the fungal infection. It's also additional factors like the patient's homeostasis and physiology, the pharmacokinetics, that is what the body does to the drug, as well as other drugs that the patient is receiving, and if these drugs interact with the antifungal and reduce its level or increase its level. If they reduce its level, it leads to failure. If they increase its level, it leads to toxicity, right? So, and then there are fungal factors which also influence the failure of the antifungal therapy. That is resistance, which we discussed already, or another term recently that is coming up is a concept of heteroresistance or the concept of tolerance, right? So let us look at what these things are. So what is tolerance? Tolerance in the laboratory is seen as growth inside the zone of inhibition in disc diffusion and in e-test, or supra-MIC growth on broth dilution methodologies. Now, the clinical implication of antifungal tolerance are not very clear, but a major study done back in 2014 found that patients who have persistent infection have more number of isolates that have tolerance compared to those patients who clear the infection, which means that the persistence of infection is dependent on the population of tolerant cells within the population of the fungus. So this tolerance is usually mediated by stress mediated adaptation and genome plasticity, which we will be discussing a little bit in detail in the next slides. So what is paradoxical growth? Now, paradoxical growth is a phenomenon that occurs with a specific class of drugs called as echinocandins. Here you can see two echinocandins that are tested. You have anidulafungin and caspofungin. If you look at well number 4, after well number 4, there is no growth in anidulafungin. However, you can see that there is a growth in the caspofungin till the end, the last well. Now, the drug concentration is increasing. Even with the highest drug concentration, there is some amount of growth. But some of the wells do not have any growth. So this is paradoxical growth. We do not know why paradoxical growth is there and what is the mechanism. And it is a topic for hot research right now. And then what is heteroresistance? Heteroresistance is usually a small population within the population of the cells that grow at 8-fold higher than MIC, higher than the majority of population. And it is also said that heteroresistance, if the number of cells that are present in the population, the heteroresistant cell, if the number is high, that also leads to antifungal failure. So the patient, despite being given the correct drug in the correct dose, can still not respond to antifungals. So this is what complicates the scenario of antifungal therapy when it comes to patient treatment. So how bad is it in India? How bad is the antifungal resistance problem in India? So we cannot get into all the antifungals, all the antifungals and all the fungi that cause infections in India. So we will be discussing about Candida species, Candida auris, Trichophyton indotini, which is causing big outbreak in India, and then Aspergillus fumigatus. So if you look at the data that has been given by the Antimicrobial Resistance Surveillance Network of Indian Council of Medical Research, we have two bugs, that is Candida tropicalis and Candida albicans, which cause majority of the infections, and these are relatively less resistant compared to Candida glabrata, Candida krusei, and Candida auris, which are known to have some amount of resistance. Now, if you look at Candida glabrata, you can find that the resistance to the azoles, that is the advanced azole, voriconazole, is around 40%, which is very worrying. And the resistance to echinocandins, which are right now the ultimate group of drugs for treating the infections due to Candida group of fungi, it's somewhere between 20 to 30%, which is again very worrying. Now, if you look at Candida auris, you find that the azole group of drugs have hardly any activity. Only 2.7% of the bugs that were tested from across India were sensitive to fluconazole, while only 14% of them were sensitive to voriconazole. If you look at the echinocandins in the Candida auris, we still have good activity, but caspofungin, again, 30% of the isolates are resistant to caspofungin. Looking at Candida krusei, we find that almost 40%, around 40% of them are resistant to both azoles as well as echinocandins, which is a big issue. However, if we look at the trends, we find that, as I said, these are the commonest agents and these have relatively less resistance compared to Candida glabrata and Candida krusei. And when it comes to Candida auris, it's very severe or very grave. We need to act fast, and we need to act now and act fast in regards to Candida auris. If you look at the drug-resistant dermatophytosis, terbinafine is a drug that was given to treat dermatophytosis, and it was said that around 17 to 75% of the isolates are resistant to terbinafine, while around 25% of isolates are resistant to itraconazole, which again is a huge number. Now, looking at Aspergillus, we find that in India, Aspergillus flavus, around 30% of Aspergillus flavus does not respond to amphotericin B, while only 48% of Aspergillus fumigatus responds to amphotericin B. However, the good point is that the drug that is the drug of choice for treatment of invasive aspergillosis, that is voriconazole, shows excellent activities in both Aspergillus flavus and Aspergillus fumigatus. Now, if we look at the mechanisms of resistance, it is mainly of two types in fungi: intrinsic and acquired. We are not worried about the intrinsic resistance, because if we know that this fungus is intrinsically resistant to this antifungal, we upfront do not give that drug to the patient. However, the scenario is complicated if the bug has recently acquired resistance. And the difference between, while the pathways for resistance seem to be the same, in primary resistance, the bug does not acquire new mutations in the presence of the antifungal drug, and the mechanisms broadly are due to drug transporters which throw the antifungal from within the fungal cell to outside, and then due to target incompatibility, which happens secondary to mutations, or target expression overexpression, which we shall be discussing in detail later. So how fungi develop resistance? Now, if you look at this, you see that there is azole, there is a gate. You can consider this as a filter or a gate. If there is the antifungal drug present, then that antifungal drug tends to choose the isolates that have, it will kill off all the sensitive isolates, and only those isolates that are resistant to it survive in the next generation. So the need or the essential part for any fungus to develop resistance is the drug. So the drug itself is an inducer of resistance, and this can be present in environment for aspergillus, that is in terms of environmental fungicide, or for the yeast, it is overuse in the hospital. Now, fungi can develop resistance while reproducing asexually or sexually, or they can also develop resistance parasexually, that is, which happens following hyphal fusion and mitotic recombination. All of these in the presence of drug, either in the hospital or in the environment, lead to selection of resistant clones of fungus. So I told about tolerance. Now tolerance is coming in big way as a cause of antifungal resistance. What causes tolerance? Now, what causes tolerance is genome plasticity. What is genome plasticity? Genome plasticity is that in the presence of the antifungal, the fungus will duplicate its chromosomes. For example, Candida albicans duplicates the left arm of chromosome 5, and this duplication results in increased number of ERG11, that is lanostirol demethylase enzyme, as well as increased amount of TAC1, which is a promoter for the drug formation. So in the presence of the increased number of transcript of these genes, there is more production of lanosterol demethylase, which produces ergosterol, and it is because ergosterol is produced, the azole group of drugs are not able to act. The other mechanism that has been described is by stress response. So there is a population of cells that do not metabolize or slowly metabolize, and when they slowly metabolize, the fungus, the antifungal drugs do not act. And then in that group of the fungi, the stress response elements like chaperone proteins, HSB90, they will change the structure of the ergosterol, and other enzymes that are involved, that are inhibited by the antifungals, they will they will make these enzymes more active, and that prevents the killing of the fungal cells by the antifungals. And so a new stress response element, that is RTA2, has been found to be a cause of stress, found to be activated in response to stress, and this is absent in humans and can act as a target for newer antifungal drugs. Now, going further, that was regarding antifungal tolerance. Proceeding to antifungal resistance. Now, antifungal resistance to azoles is mainly mediated by either mutations in ERG11, that is lanosterol demethylase enzyme, and this can happen due to mutations in this enzyme. Now, this mutations in enzyme can be due to hotspot, and three hotspots are described in Candida albicans. Hotspot 100, if the mutations are in these hotspot regions, it is said that the Candida albicans has more chances of being resistant to the azole group of drugs. Or it can also be due to gain of function mutation in this response element that will increase the amount of ergosterol demethylase enzyme, the lanosterol demethylase enzyme, to be found. Right. So hotspots and mutations in the ERG11 gene that codes for lanosterol demethylase enzyme have not only been described in Candida albicans, they have also been described in other fungi like Candida auris, Candida krusei, Candida tropicalis, and these are said to have led to resistance. So mutations in the ERG11 gene that results in the changed configuration of lanosterol demethylase enzyme, which does not allow the azole group of drugs to bind it, leads to resistance. So the next mechanism is drug efflux pumps. Now, drug efflux pumps are of two categories, that is the major facilitators and the CDR group of drugs. Now, the efflux pumps, the number can be increased by genome plasticity, that is, there is a trisomy, more number of transcripts are present, and that leads to increased expression of these drugs. Or there can be a mutations in the transcriptional regulators that makes them develop into a hypermutator type or a gain of function substitution, resulting in increased expression of these pumps that throws out the drug from the fungus, inside the fungus, to outside. So this does not allow the azole group of drugs to achieve adequate concentration and kill the fungus inside. Right. So finally, and then this is the FKS1 and FKS2 gene that codes for beta D glucan enzyme, and mutations again can result in mutations in this gene can result, or in its congener, that is FKS2, that is present in Candida glabrata. Mutations in the hotspot genes can result in resistance to change in the structure of FKS1 or FKS2, can result in resistance to the echinocandin group of drugs. Now, coming to the nucleoside analogs, the one that is available clinically is 5-flu cytosine. Now, 5-flu cytosine is transported inside the cell by use of FCY2 and FCY21 and FCY22 gene products. Inside the fungus, it is activated from the prodrug to a toxic drug by enzyme that is coded by FCA1. Now, mutations in FCA1 or FCY1, FCY2, or in any of the pathway enzyme genes can lead to resistance against flu cytosine. What we need to remember is 5-flu cytosine is a is a drug that develops resistance fast, and it is never recommended to be used alone. It was previously used mainly in the treatment of cryptococcosis. Now with multi-drug resistance Candida auris, it is also used in treatment of Candida auris. Now let's move towards Aspergillus. Whatever we discussed was common to the yeast. Moving to Aspergillus, Aspergillus is unique because Aspergillus mainly occurs not as a hospital-acquired infection, but occurs as a community-acquired infection. And in the community, if the fungicides that mimic azoles are being used heavily, which is happening, can lead to development of resistance against this group of drugs, that is the azole group of drugs, due to mutations in this transcription factor, right? This can then be transmitted to humans or can be picked up by birds from the environment and can lead to dissemination of the resistant phenotype across the globe. So now, azole-resistant Aspergillus fumigatus again has similar mechanisms. There can be mutations in lanosterol demethylase enzyme. The name of the gene is different, but again, it's the same gene. If there are mutations in this gene, it results in target incompatibility, and the azoles will no longer bind to the enzyme, leading to formation of new ergosterol, which does not allow the fungus to be killed. Or and the mutations can happen outside the lanosterol demethylase gene in other enzymes that are present in the ergosterol synthesis pathway. For example, HMG1 pathway that results in panazole resistance. And in India, it was found that around 0.8% of all invasive pulmonary aspergillosis isolates are resistant to azole group of drugs. It can also happen due to a mutation in the promoter region that results in overexpression of this enzyme. And because there is an overexpression of this enzyme, the bugs can survive in the presence of azole. It can also, some isolates do not have mutations in either this the CYP51 gene, or they do not have the mutations in the transcriptional regulator factor or regulation element, but they still can have developed resistance, and that is because of the efflux pump mechanisms and mutations that leads to increased activity of efflux pump have also been described in azole resistant Aspergillus fumigatus. This is the map of the world, which shows that many countries are involved and India is also involved. And what is interesting is that in India, we have environmental Aspergillus fumigatus that has never been exposed to in the clinic. The patients get exposed to azole group of drugs, but India also has environmental Aspergillus fumigatus, which is resistant to this group of drugs. So coming to dermatophytes, in dermatophytes, we use the mechanism of resistance to itraconazole or the azole group of drugs is the same, while the additional drug that is used, that is terbinafine, the resistance to it occurs because of mutations in squalene epoxidase enzyme that results in a change in the structure of the enzyme, which does not allow the drug to bind to this enzyme. And several mutations have been described to explain the reduced affinity to the drug. Now, that was regarding antifungal resistance. We know that the scenario is grave, but is there light at the end of the tunnel? Yes, there is light at the end of the tunnel. We have newer antifungal drugs, and these newer antifungal drugs, they either act at existing targets or they can act at new targets. Let's have a look at the newly available antifungal drugs. Now, ibrexafungerp is a novel antifungal that was introduced recently, and it also acts against the beta D glucan synthase, which is a target for echinocandin group of drug. Now, this antifungal, the new antifungal, is known to be active against Candida, Aspergillus, endemic yeast like histoplasmosis, blastomyces, dermatitis, and paracoccidoides, as well as it is active against pneumocystis carinii. Now, the advantage of this drug is that it is having high oral bioavailability, less drug interaction, and uh, it is it is active even at low pH, which is the perfect place for it to act in patients with recurrent vulvovaginal candidiasis. Moving on to the next drug, that is rezafungin. Now, rezafungin again acts against beta-D-glucan synthase enzyme, which is a target for echinocandin group of drugs, and it is active not only against Candida, it is also active against Aspergillus and Pneumocystis carinii. Now, Pneumocystis cerevisiae. Now, rezafungin is unique because rezafungin has a long half-life, which means that rezafungin can be given once a week. So it is an ideal drug for treatment of patients who have been discharged from the hospital, and you can call them to give injection weekly, or it can also be delivered as orally. So it's a perfect drug for outpatient antifungal therapy. The next one is fosmanogepix. Now, you see that the list is huge. In addition to Candida and Cryptococcus and Aspergillus, it is also active against a lot of fungi that are known to be resistant, like Fusarium, Pseudosporium, Lomentophora, Racemsonia, etc., Scopulariopsis, etc. Now, this is a new class of drug that acts against a completely new target, that is GWT1, that is that inhibits the formation of GPI anchored proteins in the cell wall, and this drug mainly acts in the endoplasmic reticulum and results in damage to manoproteins and has an extremely high spectrum of activity. The next one is opalconazole, a new drug that is active against the lanosterol demethylase enzyme, and it targets the CYP51A1 enzyme, and it is active against Candida, Aspergillus fumigatus, Aspergillus flavus, Aspergillus terreus, and rhizopus, a cause of mucormycosis. And the unique feature of this drug is that it can be given in an inhaled formulation in patients with pulmonary mycosis. The next drug is olorofilm. It has the same spectrum of activity as fosmanogepix, but except that it is not active against Candida. Now, another new drug that has come is oteseconazole, which is active against Candida and has been approved for treatment of vulvovaginal candidiasis. Now, another group of drug that was previously discussed a lot was amphotericin B. This was our one-stop solution for all sorts of fungal infection. Now, amphotericin B, as you all know, is a polyene group of drug that acts by creating pores in the cell membrane as well as increasing the number of oxygen-free radicals in the cell. Now, the problem with amphotericin B was that amphotericin B is extremely toxic, and amphotericin B can only be given intravenously. It had no oral formulation. Now, this is a new formulation which is called as uncochleated amphotericin B, which is amenable to oral formulation. Now, how is this active is that there are materials called as cochleates. Now, these cochleates are scaffolds that turn on themselves in the presence of high calcium levels, and they open themselves up, releasing the drug in low calcium levels. So the amphotericin B is put inside these cochleates and given orally. It is picked up by the intestine and then picked up by macrophages, which usually deal with the fungi. And inside the macrophages, because there is low calcium concentration, the scaffold opens up, resulting in delivery of calcium. and this results in killing of the fungus. Now this, as I said, is oral, it is stable, and it is non-toxic compared to the older formulation which had an IV formulation, and it is active against Candida and Cryptococcus. Now, moving on, of these drugs that I discussed, almost all of them are in clinical trials, and we will discuss some more that are not in clinical trials. Now, this is BAL2062. It's a newer antifungal drug which is active against Aspergillus species, including the azole-resistant strains, Fusarium, and Candida glabrata. Now, how does it act? It is transported inside the cell by a transporter protein called as CYT1. Now, CYT1 is, because CYT1 is not available in the humans, it is not toxic. Right. CYT1 is the same transport that also transports cefdierocol drug inside the bacterial cells. So this is a novel antifungal drug which is right now undergoing the preparedness, is now preparedness for human clinical trials. Right. The new one that is erratic oral bioavailability and the SUBA formulations of itraconazole. This is an electron microscopy of both formulations. The normal itraconazole, you can see it is looking like needles, while this is looking like beads. Now, this increases the bioavailability when it is taken orally. It increases its bioavailability, and that increases the chances for treatment. And FDA has approved this group of drug for blastomycosis, histoplasmosis, and aspergillosis. Now, Indians, as we are, despite not having approval for it, there was a paper to use it in drug-resistant dermatophytosis, and a paper was also produced in the Academy of Dermatologists, a poster, and people have used this in successful treatment of recurrent recalcitrant dermatophyte infection, which I was talking about. So the alternative approaches is to look at the natural products. Now, marine environments and terrestrial plants can be a source of newer antifungal drugs. For example, marine and environments have been known to be a source of alkaloids, polyketides, peptides, and these, for example, zemamidine, thionelamide, these bind to ergosterol and have been found to be active against Candida albicans. Similarly, terrestrial plants are a source of essential oils, and these can inhibit the biofilm formation and cause membrane disruption, again active against Candida albicans and flavus. So there is a demand for looking at newer antifungals because the antifungal armamentarium is limited, and this is a research opportunity for all the people who wish to look into antifungal drug development. Now, nanoparticles, that is, have been available for long, and nanoparticles can kill fungi by multiple mechanisms. Though they are available for long, their translation into the human trials is less because they are known to be toxic to humans, right? But the advantage of using the nanoparticles would be that nanoparticles are known to be active against biofilms, which is an intrinsic resistance mechanism and a very important factor when it comes to development of infection due to indwelling devices in human infections. So the other way of looking at it for drug discovery is drug repurposing. And how drug repurposing is done is via this pathway. You first mine the pathogen genome data. You look for orthologs to known drugs. You do a homology modeling, and then you do an in silico ligand preparation and docking, and finally you do experimental validation. What are the tools for data mining and pathogen genome data? You have the Broad Institute Genome Database. This will give you the whole genome sequences of the fungus for whom you wish to see if the available drugs can act against their orthologs. And orthologs can be found out by OrthoVen server. What will be the source of the drugs. It can be drug bank, therapeutic target database, and it can also what genes can be found out by the DEG database. Now, homology modeling can be done by these softwares, that is Swiss model software, protein database, bank, Ember 99, and PubChem server. While ligation, preparation, and docking can be done by these servers. I am not a bioinformatician. This is not my area of work, but this is broadly the way it is done. Finally, experimental validation can be done by using CLSI or UCAST or its modification. So what is the source of the drugs? We can completely find new drugs, that is like, or prevent that and look at the available group of drugs and screen them. So what are the available group of compounds? The available group of compounds are available in drug compound libraries like the malaria box, the pathogen box of the malaria venture itself, or Prestwick chemical libraries have a vast number of compounds which can be tested against this drug resistant fungi. And the other way is, and after doing it, there was one trial, that is the defeat trial, where they used defarasirox along with amphotericin B. In animal models, it had shown good response, but when it was applied to humans, it failed badly. However, recently there has been a preclinical trial in the animals, not a preclinical trial, an animal study in which pioglitazone, that is an anti-diabetic drug, which was used in conjunction with amphotericin B, and this led to reduction in the fungal load and helped in early clearance of the fungus in the mice model. Whether it will make to human clinical trials and whether it will be successful is a matter that needs to be investigated by further research. Another anti-parasitic drug, that is niclosamide, was also found to have antifungal effects by preventing filamentation of the fungus, by preventing biofilm formation, and by also affecting the fungal growth and survival. Now, this is the approach of drug repurposing. As I said, drug repurposing can be done by looking at the genome, finding out the gene or the protein orthologs, in silico finding out if the drug that you have chosen can bind to the active site or can inhibit the target of these molecules. And once you find out it in silico, try and experimentally validate it on wet lab, and then take it to animal experiments. And if there is adequate experimental data, it will make out for human trials. The other way is the immunological approaches. Now, immunological approaches have been viewed very, very badly in medical mycology, because the people who develop these infections are people who are already having a very weak immune system. So you can't expect a person with very weak immune system to mount an immune response. So there, the adaptive D cell, dendritic cell transfer, or T cell transfer, these approaches were found to be successful, at least in animal models. However, recently people are also working on developing vaccines for various invasive fungal infections like invasive aspergillosis, candida, and other group of drugs. And recently, cellular immunotherapy and molecule-based immunotherapy is also being looked into, and these are avenues of hot research right now, because we know that the antifungals are not going to work. So let us take care of the fungus by enhancing our immune system. So what are the cellular immunotherapy? The CAR T therapy, the CAR NK cells, and the new molecules like soluble pattern recognition receptors, the immunoglobulins, or the members of innate immunity. These have all been tried and tested, at least in the petri dish or at animal models. So that is also an area of work which is now slowly expanding because of this small armamentarium of antifungal group of drugs. New targets. I spoke about the GPI anchored protein when I was talking about the newer drugs. So other than that, there is chitin synthase. Nicomycin was a was a antifungal that was used, that is available but not used rampantly, and it has been found to be useful in conjunction with echinocandins for treatment of endemic fungi, that is histoplasmosis, blastomycosis, so on and so forth. And now there is this these genes, are the products of these genes, that is DFG5 and DCW1. These are stress response. These are elevated in response to stress, cell wall stress, and these also are involved in hyphal morphogenesis. And ultimately, what they do is they result in activation of the chitin synthase enzyme. Now, chitin synthase is not present in humans, which means that if you target this, then the human toxicity is going to be less. So, this group of this group of enzymes is something that is being looked into for development of drugs. Now, other strategies is use of AI. AI is everywhere these days, and people are saying that the big data that is generated by whole genome sequencing, by the uh, drug screening methodology that I talked in the previous slide, it can be made easier by applying AI and machine learning, and this approach can help optimize drug candidates as well as predict resistant mechanisms in the bugs whose genome has been what should I say, sequenced. Now, this approach was used in Candida auris, and it is available for uh, other bugs as well. However, the problem is the type of data that you use to train these algorithms. So garbage in, garbage out. If you don't have good data, for example, you have a colonizing isolate from a sick patient, and this colonizing isolate is not the cause of the disease. If you sequence this and you apply algorithm, obviously it is not going to give you the results if you would have sequenced an isolate that actually came from the site of infection and was causing the infection. So other strategies are anti-virulence approaches. We know that fungi communicate amongst themselves by using quorum sensing mechanisms. Quorum sensing is not available in human beings, although its evolutionary pathways, other cytokine quorum sensing, but the enzymes or the molecules that are involved in fungal quorum sensing, like farnesol, penicillic acid, these are not available in mammalian cells. So you could target the quorum sensing genes or quorum sensing enzymes, and this could potentially be a source of newer antifungals. Diagnostic advancements definitely help in reducing the burden, at least in the hospital, if not the environmental azoles, because the earlier you diagnose that the infection is because of this fungus, you can stop unnecessary empiric therapy, and that will result in less load of the antifungal drug. And we have several new technologies like MALDI-TOF, BioFire, T2, Candida assay, and next generation sequencing, in addition to biomarkers that help in rapid identification of fungal infection and help in something called as antifungal stewardship. I will not talk about antifungal stewardship because it is a topic in itself, and this can be discussed on some other day. But the point is, if in a patient who is having fever in an intensive care unit, you don't know if the fever is because of bacteria or because of fungus. And till the time the results are available, the clinician starts the antifungal drug upfront to prevent the patient from dying. So if the result of the antifungal susceptibility is available earlier, you can remove the antifungal if there is no fungal infection, or you can add antifungals only if there is evidence of infection. So this will remove that azole stress, that antifungal stress that selects antifungal resistant clones. So this is the principle of antifungal stewardship, and we will not discuss it in further details. So how can this is a proposed mechanism by Matthew Fisher and others. It was published in 2022, and they said that you can do surveillance and resistance detection and tracking by involving citizens. So the citizens can mail the material from the environment to centers which are equipped with ability to sequence the genomes, and this is then uploaded onto the cloud where phylogenomics, surveillance, and transmission tracking can be done, and we can do a real-time tracking of resistance against the bugs. This is very ambitious, and it sounds very good. But who will fund this kind of a study? That's a problem when mycology itself is underfunded, and the lead body of the country is planning of shutting down some advanced mycology center. This is something that needs lobbying, or that needs to be told to the policymakers, told to the funding bodies that you should not do this. This is not the time to shut down centers. Rather, this is the right time to support them. So globally, I already spoke about One Health. Fungi, I told there are some fungi which can cause species extinctions. And though we have not yet encountered something of that sort in humans, it is very well possible that we may find something of that sort in future. And if we have to be prepared, we have to look at the effect of the fungus on the environment, on humans, and in the animals as a One Health approach, and this can prevent development of further infections. So what is the way you can do it? You can do it by doing environmental antimicrobial resistance surveillance, wherein you stop the usage of fungicides that share the same mechanism of action uh, to the antifungal drugs that are used in the hospital. In the hospital, you do antifungal stewardship, you do molecular resistant diagnostics, and then at national level, you deploy surveillance centers, you support advanced mycology centers, and you do a surveillance in these centers. So this can probably reduce the level of antifungal drugs. So what is the future? The future is funding and investment in mycology by both academia, funding, and investment. So investment can be in terms of money, investment can be in terms of brain power. So academia needs to take a look at mycology. Academia needs to invest more in medical mycology, more in environmental mycology, so that we are out with new drug targets, new mechanisms, and we are able to take care of this epidemic of antifungal resistance. And then in the clinics, there is need for robust treatment protocols. We have treatment protocols for Candida, Aspergillus, Cryptococcus, but we do not have proper standard treatment protocols for phaeohyphomycetes, for Mothorella mycetomatis. So there is also need to look at synergistic drug combination, how they can help. And then definitely there is an avenue for more research in antifungal drug and diagnostics. Antifungal drugs because they will directly be used in treatment, diagnostics because they will reduce the time to diagnosis and cut down the duration for empiric treatment. So concluding my talk, there's an urgent need for innovation in terms of antifungal drugs and antifungal and fungal diagnostics. And this can probably be achieved if there is a culmination of basic scientists, of computer data scientists, and clinical scientists. And this seminar should or this webinar should, make our younger generation and the people who are in academia and the people with the links to politicians as well as to the funding agency to lobby for more funding and more interest in the medical mycology as well as mycology in general. So I will conclude this talk by saying that we are a reference center for mycology medical fungi, and if any one of you is taking care of a patient with a fungal infection which you are unable to identify or unable to do its antifungal susceptibility, please contact us. If you feel that you have a outbreak of fungal infection and you have kept those isolates, please contact us. We'll be happy to type them for you. So additionally, there we will be advertising a seat in mycology in the coming session, and this will be open to students who have their fellowship, by either DST, DBT, or ICMR. They can come and join our group and can work on development of diagnostics or any other aspect of medical mycology. So this is we are here to help, and these are our various links. And thank you very much, Dr. Damodar. Uma madam.
Yes, sir. Thank you, sir. That was a very elaborative session which covered almost all the aspects. But one thing I would request from Dr. Vinay, here there are researchers as well as the academicians who are working even on the fungal component in the agricultural sector, like a plant pathology. So I just want you to share a few words prior we start with the QA sessions, the connection between the fungi, example like Aspergillus and Fusarium, which is in a plant pathology where they are also playing a major role over there, as well as the Aspergillus and Fusarium, which also is connected with the medical mycology, especially in the hospital sector.
So regarding the intersection, One Health is
Yes.
something that
Yes.
is an intersection of medical mycology and veterinary mycology and plant mycology.
Yes,
As well
yes.
as it's an intersection of environmental sciences. Now, the thing is that previously we were working in silos. The people working in medical mycology used to contact taxonomists, used to contact plant pathologists if they could not identify a fungus, because that was your forte. However, in agriculture, because food security is a big issue right now, and a lot of antifungals are used in the agricultural sector, and these antifungals have the same structure as the antifungal drugs that are used in the clinic. So if they are persistently present in the environment, then they will result in development of resistance, and the person who is at risk for fungal infection will be infected by them from the environment and then land to the hospital. And when the clinician gives the antifungal, it won't work. So that's the cycle. And regarding fusarium is a big plant pathogen. It's a problem to people who are treating fungal infections because it's multi-drug resistant, and it's very difficult to treat. And there's not much of work with regards to India as to what's there's need for more medical mycology research in India, more concerted efforts, not only amongst medical mycologists, but also the plant pathologists and the taxonomists, to bring forward the numbers. Actually, where we stand is we don't have the numbers. Whatever numbers I presented today are from mathematical modeling. We don't have surveillance. Fungal infections were never a part of notifiable diseases. It was only with the COVID-associated mucormycosis epidemic that the government of India woke up to the fact that even fungi can cause infections in normal people and made it notifiable. So with regard to data and with regard to opinion, I can just say I have opinion, but whether my opinion is based on scientific data, this is just my opinion.
Okay, thank you, sir.
With respect to India.
Yeah, thank you. I think so with this, we have a lot of researchers, young researchers and the academicians. I think so here we can build up like a collaborative effort is needed to bridge a gap between the hospital or environmental. As you said, the One Health concept is very much required at this moment. So with this brief up now, I request Shenoy sir to make the session open for a Q&A.
Before starting, I again want to reiterate, I am the new kid on the block. Okay, so I will try my best to resolve your queries. And again, I want to reiterate, there is not a lot of data from India. with respect to the disease prevalence. What is we simply don't have the data. So if your questions are related to the prevalence and those things, I may not be able to answer it, but let me try to answer it at the best of my abilities. Thank you.
Thank you.
Thank you. Thank you, Dr. Maheshwari, and thank you, Dr. Vinay, for the wonderful presentation. I really enjoyed listening to you, and I'm sure the audience has learned many new things, actually. If ladies and gentlemen in the audience, if you have any questions for Dr. Vinaykumar Hallur, please use the raise button in the Google Meet platform, or you can put your questions in the webinar WhatsApp group. Dr. Prakash Halami, welcome to the program. Do you have any questions?
Good afternoon, sir.
Good afternoon, sir.
First of all, I congratulate Dr. Vinay for an excellent presentation. In fact, I work on AMR in bacteria, and this was really insightful information that what mechanism which we see the resistance, excessive drug resistance or multi-drug resistance is totally different here than what we see in the bacteria. Again, with reference to the cross resistance and inducible resistance. So what I see here, most of the targets are with the cell wall associated, the antibiotics which you use. How actually antibiotic resistance will differentiate between virulent and non-virulent cells? Because the virulent are the one which are actually a cause of concern with reference to the infection. I hope I am clear with my question.
Sir, your question is clear, but the status of virulence versus non-virulent is actually in a dilemma kind of thing, because with respect to Aspergillus, I can say that during the 90s there were studies in which they found that certain clinical isolates of Aspergillus fumigatus are more virulent compared to environmental isolates. And then there are anecdotal studies that clinical isolates are more virulent than environmental isolates. That is with respect to the common fungi. I am not talking about the really virulent fungus like Histoplasma capsulatum, blastomyces, which cause infections in everyone. The cause of concern in medical microbiology is that our set of patients are immunocompromised, and it is those fungi which have low virulence potential also can cause infection. So even amongst them, whether clinical isolates are more virulent, whether environmental isolates are less virulent is something, it's a matter of debate, it's a matter of research, and for several fungi it is still not clear that are there actually any really more virulent strains compared to the clinical to the environmental isolates. So this is the status with respect to the virulent versus non-virulent fungus, sir.
Wonderful. Just one more. Actually, the antifungal, fungicide which you were telling, which you use in agriculture, also is a reason for developing a resistance in fungi, right?
Yes, sir.
So how is that cause of concern? Because like fungicide is very commonly used in agriculture for plant protection,
Sir,
is it something like a cross resistance? Is it something like inducible resistance which we see in a bacteria?
Sir, what happens is you have the drug-resistant fungi in the environment,
Yes,
and the patient is roaming around in the community. A patient with less immunity is roaming around in the community, picks up that fungus, which is already resistant to the available drugs. He comes to the hospital, we give him the drugs, he does not respond to it because the environmental isolates which can cause infection in him have already become mutated and acquired resistant. That is because the same class of drugs that are fungicides are also the same class of drugs that are antifungals. It is the same class. The name is different and the structure is little bit different here and there. For example, you know,
Okay.
sir, penicillin. Penicillin has
Yes.
the same backbone in all the penicillin beta, in all the penicillin group of drugs, beta lactam group of drugs. But the side chain is different. There is addition of something. So like that, the azole group of drugs, the fungicide in the environment has the same backbone. The one that is used in humans is less toxic to humans compared to the one that is used in the environment. So that is the cause of concern. And medical mycologists in Europe are pitching for banning the use of these fungicides in the environment, because
Okay.
the loss of life, the loss of human life is of great concern there. They want the agricultural industry to use other fungicides than the ones that have similar mechanism to the antifungals used in humans.
Thank you. Thank you. Just one last general question. Actually, I hope it is okay with the organizers. No, we will usually have a large amount of fungi in our gastrointestinal tract, which may be most of them may be harmful only. Can we use a strong antifungal antibiotics and get rid of them? What is your opinion?
Sir, I can just give an opinion. I cannot say that I am an expert on this, but in general, there is something called as a microbiome. Okay,
Exactly that concept.
the gut has a microbiome, and the components of the gut microbiome prevent infection from other microbes. Right. So Candida is a normal part of our microbiome. Fine. So what function it is doing, Candida, Malassezia, and many other fungi, they are part of microbiome, and it's still not clear, because in normal human beings they don't cause disease. Why do you want to take out Candida? Or why should I be worried about somebody who is walking, talking, and healthy? Why should I give him some antifungal and take it out? I would be interested in taking it out only in a patient who is at risk for development of fungal infection. And that is done in patients, for example, who are at risk for development of hepatic encephalopathy. There is a syndrome or disease called as hepatic encephalopathy. In that group of patients, it is given. So not a common practice though.
Thank you. Thank you very much.
Dr. Vinay, one of the persons in the audience wants to know what is the role of probiotics in combating fungal resistance. I think you partly answered that in the previous question, right?
Yes, sir.
Okay, thank you. Dr. Yusra wants to know, if we want to investigate resistant genes at the molecular level, what would be the most affordable and effective method for detection and analysis? This is from the Mycology WhatsApp group. If you want to answer right now, you can do that. Otherwise, you can answer later too.
So Dr. Yusra, there are two approaches. One is the, if I will just repeat the question to confirm if I have understood it right or wrong. So you want to know the most economical approach to characterize resistance. If that's your question, the most economical approach to characterize molecular characterization of resistance would be performing PCR of the resistance gene followed by its sequencing. Considering that you would have already done a phenotypic characterization and know that this bug is resistant to this class of drug, you would choose the common mechanisms associated, do the PCR for that gene, and then sequence it. Or the more expensive and what should I say, a less selective approach would be doing NGS, that is next generation sequencing. You do de novo sequencing, you look at all the enzymes and you look at all the mutations. But for doing this, you need to have an excellent bioinformatics team. You should have somebody who has worked with fungal genomes and knows how to find out the resistant genes in that pool of massive data.
Thank you, Dr. Vinay. One more question. If you ask somebody from the Microbiology WhatsApp group, if we are designing a nanoparticle or nanopolymer as a carrier for our lead compound antifungal under study, what all are the major things to be considered? Do you want to answer this question?
This probably, if I understand this right, there are two parts to this question. One is a proof of concept study, and second is evidence of safety that has to be given to the regulatory bodies prior to entering preclinical trials. So if you're wanting to do a proof of concept, first you design your nanoparticle. In the lab, you see if it is active against the said fungus or the group of fungus. And then you do something called as preclinical animal studies. You take the animal model of fungal infection, you give your nanoparticle, and then you look whether there is a response to the nanoparticle that I've given in the animal model of the fungal infection. You find this, then the next step is a big one. You have to there is the Drug and Cosmetic Acts of India 2017. It gives a entire list of animals, cultures that have to be put, and toxicity assays that have to be done, and then once you present all this data to the regulatory body, it goes to preclinical testing. So that's the flow. So first a proof of concept, and then regulatory approval, and then preclinical trial.
Ladies and gentlemen in the audience, if you have any more questions, you may please. Dr. Teresa, wait, let me. No, not Dr. Teresa. Anyway, Dr. Vinay, I think there could be some more questions in the groups, and you are part of some of these groups, so you could answer. Let's see if anybody wants to ask any more questions. No, looks like there are no more questions from this audience in the Google chat.
I think Abdul Razak Mustafa has raised his hand.
I think we can take it in the webinar WhatsApp group later. I will put you in that WhatsApp group. I would request Dr. Sunil Kumar Deshmukh, the president of the Association of Fungal Biologists, to share some comments. Yeah.
Actually, what is my opinion of medical mycology is that it is restricted, the expertise is restricted to few labs only. So this is one reason that people are not taking interest, that they are scared of medical mycology. I don't know. Secondly, the taxonomical part is actually ignored. Thirdly, that like Chandigarh, Dr. Chakravarti and those group is working. If you see Vellore, people are working. But number of experts in medical mycology is less as compared to European countries. Secondly, the person who is in medical science is not interested in working in medical mycology. They are more interested in bacteriology and others. That is another reason. And the person who is working with botany or microbiology, they feel that if I work with medical mycology, where I will get the job. Because getting the job in medical science, they will ask whether are you MBBS or MD, then you will get job in medical college, otherwise you will not get. This is another reason of not people are opting for medical mycology. Secondly, about natural product in particular, initially Hex Pharmaceuticals, Piramal or RRL Jammu, they were working. But nowadays, if you see the publication on antifungals from natural product, particularly from India, hardly you see 1 or 2 papers. So that is the preliminary data. So getting the antifungals from India, particularly from natural product, chances are very less, because first process is getting the antifungal, then you have to do pharmacokinetics, then clinical stage, because these are the lengthy stage, lot of money is involved, so people stop after getting, showing that it is antifungal property. I was working with deoxymelanocanine with Hex Pharmaceutical 1992, we published first paper. Then aminocandine was developed, showing good profile, but Vericor Pharma, they stopped working. The reason I don't know, even after passing the clinical trial phase 4. So there must be something related to the marketing thing, so they have stopped. So chances of getting antifungals, particularly from India, that to echinocandry class will be rare. And if we want to involve, we have to develop interest in the medical science people as well as the mycologist who is working on taxonomy will be there. So I think we have to educate the people, create interest in the youngster to work in the field of mycology, first in mycology, then in medical mycology. I think yeah.
Thank you, Dr. Deshmukh. Dr. Uma Maheshwari, can you please summarize the today's proceedings? Thank you.
Yes, sir. First, I would like to thank Dr. Vinay on behalf of AFB, AMI, Mysore chapter, as well as from JSS here, for giving such an elaborative presentation. Right, he started from what exactly the fungal resistance is, and he covered almost the topics which are very much interesting, like the Candida, Aspergillus, and Cryptococcus, their mechanism of resistance, mechanism of action in showing the drug resistance, and how actually the resistance was in turn, the techniques which are used to detect the resistance, and the newer approach, which is very much needed, like the immunological approaches, how exactly they are playing a role, because especially these fungal infections usually arise whenever there is a drop in the immunity system. So the approaches which we need to unravel in future, that's the main thing, I think. So the young researchers as well as the academicians as well as the scientists should need to think. And definitely I agree with the statement, like the press and the social media is very much required to drive the attention of the researchers who are working mainly on a bacteria, and even the young researchers should take more and should have a concern more on the fungi. So usually, whenever the work starts with the plant pathology part, I've seen like most of them, they go with the drug molecule and phytomedicine. Whenever they try, they'll just work with the antifungal activity and they just leave that work. So I think so here we need to have a more concern, because from an East point of view, usually they work with the Candida albicans, as well as from the fungal point of view, the Aspergillus as the fusarium. Most of the phytopathology people, they'll work on this, and the pharmacy people also work on with the antifungal component. So there is a much need in the microbiome component, as sir suggested. There's a work going on with a lot of bacterial work. So other thing is now the microbiome needs to be addressed. So with this, I also thank Dr. Vinay for having given us the invite that we can approach him where if there is any. With this platform, I request those who are having interest and who can collaborate. So also shared a slide where there was a mail ID as well as the contact details. I think so the participants can make use of it. And definitely, Dr. Vinay, I'll be in touch with you. And thank you. And that was a very elaborative presentation. And adding to this, yes, politicians, funding agencies, academicians, all these people need to take an interest on mycology. Thank you once again. And now I request Shanoy sir.
Thank you, Dr. Uma Maheshwari. Ladies and gentlemen, on the behalf of the organizing committee, first of all, I would like to thank Dr. Vinaykumar Hallur for spending his precious time with us and the knowledge of antifungal resistance and medical mycology with us. He has been a wonderful, energetic, and positive, speaker. That we see him we see his interactions every day in Microasia WhatsApp group. So thank you, Dr. Vinay, for your kind support to this kind of initiative. And I'd like to thank our collaborators. The collaborators include the JSS Academy of Higher Education and Research, Mysore, and AMI Mysore Chapter, Association of Fungal Biologists. From the JSS, I would like to profusely thank Dr. Uma Maheshwari. I think this program wouldn't have happened without her active participation. She was the kind of initiator of this particular event. Thank you, Dr. Uma Maheshwari, for your kind support and collaboration with us. I would like to thank the Dean of Research from JSS, Dr. Prashant sparing some time from his busy schedule to participate in the inaugural session. He gave some good insights about the medical mycology, medical microbiology. Thank you, Dr. Prashant. And from the AFB, I would like to profusely thank our president, Dr. Sunil Kumar Deshmukh, and he has been quite encouraging and supportive of this kind of interdisciplinary, beyond fungal taxonomy kind of on webinars, especially leading towards biotechnology, leading towards applications to the common people. Thank you, Dr. Sunil Deshmukh, for your kind encouragement and facilitating these collaborations. And I would like to acknowledge the important roles played by the office bearers of AFB in this for this event. And I'm very grateful to this audience. We received around 700 or 800 registrations, and I expected around only 200 people today. Looks like we have got around 300 people in the audience at one time. Thank you very much for your enthusiasm, being patient with us, and patiently listening to Dr. Vinaykumar Hallur and participating in the question and answer. I have already shared the feedback link in the chat box. If you are key, I very much appreciate we very much appreciate your feedback. Please give your feedback. And if you want the certificate of participation, you will be given a participation certificate from the JSS. Thank you very much. I think with this, we sign off, Dr. Uma and Dr. Vinay.
Yes, sir. Thank you.
Dr. Sunil Kumar Deshmukh, with this, we sign off.
Sure.
Dr. Halami wants to say something. Dr. Halami, please. Dr. Prakash Halami, you have anything to say?
No, sir. Actually, it was by wrongly I pressed, but I just thought as you have given me a mic, so I would like to congratulate and thank all my co-team for collaborating with the AIIMS Mysore chapter. Thank you, sir.
We would like to have more interactions and more collaborations, more events in association with the AIIMS Mysore chapter. Not only Mysore chapter, with the AIIMS central body.
Definitely, sir.
Thank you for participating in today's event and interacting with us. Thank you very much. With this, ladies and gentlemen, we sign off. Have a good Saturday wherever you are. Thank you. Bye-bye.
Thank you, sir. Thank you.
Thank you, sir.
Namaste.